My name is Dr. Dayan Goodenowe. Most of you will first come across my name in relationship to my research in Alzheimer's disease, and specifically the role that plasmalogens, an essential membrane phospholipid, plays in the etiology of Alzheimer's disease. You will find on this website and on others detailed descriptions of what plasmalogens are, where they come from, how the body becomes depleted in them, and how a low level of plasmalogens can lead to an increased risk of Alzheimer's disease and cognitive impairment. However, I thought I'd take an opportunity to give you a little bit of background on myself and on the research that has led us to this point in time. Fundamentally, my background is in synthetic organic chemistry, and my first degree was in chemistry, where I looked at the designer amphetamines and their role as hallucinogens. And from there, I did a PhD in neurochemistry, where I looked at the biochemical mechanisms of drugs used to treat refractory depression, specifically combination drug therapy and its effect on different neuronal systems of the brain. From there, I started working in the pharmaceutical industry, again, focusing on neuronal mechanisms of disease, looking at acute brain injury and how we can prevent neuronal death following car accident or stroke, looking at the anxiolytics, which help prevent or reduce the levels of anxiety, hypnotics, and how they improve sleep function. And this is all done by designing molecules that can allosterically modulate the receptors and the membranes in which receptors exist. Now, in the course of research, what happens quite frequently is that when you pose a question in research and you want to design experiments to answer those questions, often technology exists, but sometimes the technology that's required to perform the experiments that you want to do doesn't exist. And at that point in time, many times scientists have to become tool makers. They need to stop doing research for a period of time to actually build equipment and technologies that are required to answer scientific questions. If you want to look at the stars, you need a telescope. And so someone has to build the telescope. And that's what happened in my particular situation. This was in the late 80s and early 90s when the genomics revolution was taken off. And that means we had the capacity to sequence the entire human genome and the genome of other organisms. And when we looked at the entire genome in its complexity, we could start looking at how certain genes have certain functions biochemically. And these genes get translated into proteins, and these proteins also have functions. But proteins and genes can be sequenced. My background is fundamentally in chemistry and biochemistry and the biochemical mechanisms that occur in the human body. And unfortunately, a similar level of technology did not exist. The measurement of small molecules in biological fluids is an incredibly complex process, and normally you can only measure certain types of small molecules at one time. And in order for me to research the total complexity of biochemistry simultaneously in research, looking at one disease versus another, or versus one genotype versus another genotype, I needed to develop an analytical technology that can measure all those small molecules. And so that's my first invention, was an invention called comprehensive non-targeted metabolomics. And what that technology allowed me to do was measure thousands and thousands of small molecules in a biological fluid. And it used ultra-high field mass spectrometry to do it. A mass spectrometer that can measure all the small molecules with high degrees of resolution and high degrees of accuracy. And that particular invention really was the beginning of a vast amount of additional research that then came from that. Because once we had that tool at our disposal, we could now measure thousands and thousands of blood samples from people from all around the world. People with colon cancer, people with pancreatic cancer, ovarian cancer, breast cancer, and compare them to people who did not have that disease, as well as look at them before surgical removal of tumors, after surgical removal of tumors. And that was in the cancer field. In the neurological field, we're looking at people with Alzheimer's disease, with different degrees of cognitive impairment, Parkinson's disease, ALS, multiple sclerosis, autism. These are the diseases now we can start studying the biochemistry at a very large understanding of the system. And what happened early on, the purpose was to understand the biochemistry of that particular disease so that we could diagnose those diseases. And as more and more biochemical information became available, it became clear that these diseases had a prodrome. And a prodrome is a biochemical environment that precedes the clinical manifestation of a disease. And so we could actually start seeing in individuals biochemical changes that were occurring before they had the disease, which was making individuals susceptible to that disease. And that became quite useful, and I filed many, many patents on the diagnosis of disease using this technology platform that I invented called non-targeted metabolomics. And so we could diagnose autism, and we could diagnose Alzheimer's disease, and multiple sclerosis, and pancreatic cancer, and ovarian cancer, and colon cancer, and so on and so forth. And what became very interesting was that the more information that we were generating across larger diversities of humans, that this information wasn't getting more and more complex. This information was starting to get simpler and simpler, and things were starting to organize into relatively straightforward biochemical systems that became dysfunctional prior to certain diseases. And that's where prodrome sciences came in. And so the prodrome neuro plasmalogen supplement was one of the first discoveries made using this technology platform. And it happened when we looked at people with dementia versus people that did not have dementia. And people with dementia had low levels of these molecules in their blood called plasmalogens. And from that low level of plasmalogens, we could understand the mechanism of how people with low plasmalogens had a high susceptibility of becoming demented. And that will be described in much greater detail in other presentations that you can see on in my published papers or videos on this website. And so once we found this decreased level, the next step was, can we replace them? And so that's where my synthetic chemistry background comes into play. So we can actually, I'm a designer of molecules and designer of chemical systems that allow us to manufacture the exact identical biochemicals that are in the human body, and we can then target them into a supplementation model so that we can restore the body's natural biochemistry to a state that is resistant to and no longer susceptible to certain diseases. And that is really the first part of prodrome sciences. Now, as time goes on, and we looked at the prevention of disease and the eliminating the future prediction of disease, was why should we stay limited to the prevention of a negative, like a disease? Why can't we look at this one step further? Can we create biochemical reserve? Is there a way that we can apply the same concept of here's a deficiency that causes a disease? So a prodrome to a disease is typically a deficiency or a dysfunction. But the opposite is also true. People with a reserve capacity in certain biochemical systems, not only are they resistant to getting a disease, they actually have higher degrees of vitality and a higher degree of longevity. So can we now take the prodrome concept to one step further and not really look at the prodrome of a disease, but the prodrome of health and the prodrome of vitality? And so that's kind of where the prodrome scan blood testing technology comes in, where we can now measure large amounts of biomarkers in the human body and start optimizing them, not in a strange way, in a very natural way. There are individuals just by random chance have optimal or reserve capacity. And so can we take that knowledge and apply it to everybody so that everybody can have biochemical reserve, and we can prevent disease, and we can improve longevity, and we can improve vitality in old age? So when you look at the initial products on our website, Prodrome Neuro for one, I hope this explanation of the kind of science that I do and the research that I'm involved in. So we have clinical trials all around the world. Colon cancer, pancreatic cancer, breast cancer, bipolar disorder, schizophrenia, Alzheimer's, Parkinson's. And these controlled trials that we do around the world are designed to get better and better knowledge of the biochemistry of disease. And then we apply that information to not only understand how do we prevent a disease, but studying the people who don't get these diseases and understand what is the optimal biochemical environment for the prevention of a disease and for the elongation of vitality. And that's what I'm truly interested in. So welcome to my research, and I hope you enjoy your quest of knowledge with me. Thank you.